We are privileged to have her chair today's session. Welcome, dear Linda ma'am. I now hand over the session to you.
Very good afternoon, everyone. Thank you, Dr. Pooja, for your kind introduction. Thank you, Teamas, for giving this opportunity to chair the session. We are privileged to have with us today a distinguished expert and industry leader, Mr. Antoine Francis, the founder and CEO of Global Plasma Supply Limited, based in London. Mr. Francis has been at the forefront of innovation in the plasma supply chain with vast experience spanning policy, operations, and strategic partnerships in the global plasma industry. He has done his Bachelor of Science in Health, in Clinical Laboratory Sciences and Master of Science in Transfusion Science and Cell Transportation Technology. And he has more than 15 years of experience in transfusion healthcare, serving a variety of roles, and he has many more achievements. Led creation and UK government endorsement of cross-border plasma supply business plans. And he has been regularly promoted within roles due to sales, overperformance, leadership, and innovation. Has been recognized by international and European bodies for scientific and operational standards. Has achieved 96% market capture in Lebanon medical device and consumable segment. And he has been awarded the Best Team Spirit Award by multiple times by AstraZeneca. With that, now I request all the participants to remain muted during the session. If anyone wishes to ask any queries, can ask towards the end of the session. I warmly invite Mr. Antoine Francis to share his insights. Over to you, sir.
Hello. Can everybody see my screen? All right. Thank you for the introduction, Dr. Linda. It's an honor to be here. I would like to say good morning in my time to everyone who's here. It's a privilege to be here and share everything I've done exactly for this plasma fractionation industry. I'm talking about the procurement and I'm talking about the fractionation and how the whole industry is now being drifted and shifted towards what we call a volume from volume to a value in fractionation. I will welcome you to the session, evolving a paradigm in the evolving paradigm of plasma fractionation.
Sorry to interrupt you. We can't see your screen.
All right, sorry. Let me see. Can you now?
Yes, we can see. Thank you.
There we go. So again, I would like to welcome you all to this session on the evolving paradigm in plasma fractionation. Historically, our field has been governed by volume-driven imperatives, maximizing plasma usage across all protein outputs. And today, we find ourselves in the midst of a strategic inflection point, a shift towards value-driven models, where market focuses particularly overwhelming demand for immunoglobulins. Now, it dictates now what it collected and how it's processed, where the investment is flowing. This presentation will critically explore the mechanisms, consequences, and future pathways to the shift. We will assess how IVIG has emerged as the central economic and clinical driver, reshaping not only production priorities, but also donor engagement models, purification technologies, and international plasma policies. The core question before us is this: how do we ensure that this transition serves both efficiency and equity, preserving the broad therapeutic potential of plasma while meeting soaring global needs? Let me go to this next slide. This slide offers both historical and biochemical overview of plasma fractionation pathways. On the left, we see the original cone fractionation schema, a landmark achievement in the 90s. Cone ethanol fractionation process allowed systematic isolation of plasma proteins based on solubility, ionic strength, and temperature and ethanol concentration, ultimately producing 5 major fractionations. Each fractionation contains clusters of functional distinct proteins, such as albumin, fibrinogens, and immunoglobulins, laying the foundation of modern plasma therapeutics. On the right, we observe how modern fractionation has evolved while cone methods remains fundamental. Contemporary plasma processing integrates multiple chromatographic steps, allowing higher yield and purity. Today, we extract not only albumin and IgG, we also go into specialized products like alpha-1 antitrypsins, coagulation factors, and importantly, like protein Cs. This process starts with either whole plasma or cryopoor plasma, highlighting the importance of upstream donors and plasma handling protocols. These processes are not merely biochemical, they are economic and ethical in frameworks, determining which molecules are prioritized, which are discarded, and which make it to the patients in need. We transition into a value-based strategies, and this molecule maps a mirror of our intentions and our blind spots. Before delving into the economic and molecular drivers of modern plasma uses, let's revisit the fundamentals. Plasma fractionation is the process of separating human plasma into its protein components, which we call plasma-derived medicinal products, PDMPs. Clinically, the core products we focus on are immunoglobulins, albumin, clotting factors, alpha-1 antitrypsins, and other specialist proteins. The origins of this practice date back to World War II, when Cohn's method was first implemented at scale to provide albumin to wounded soldiers. Today, while over 300 distinct proteins have been identified in plasma, only 15 are purified into commercial therapeutic products, a figure that reveals both our progress and our limitations. So plasma fractionation is more than separation. It's clinical triage at the molecular level, determining which proteins are rescued and which remain untapped. To appreciate the paradigm shift we're witnessing today, we must first understand what came before. The traditional approach to plasma fractionation was fundamentally volume-driven, a philosophy centered on maximizing output from every liter collected. The goal was clear: extract everything, albumin, immunoglobulins, clotting factors, ensuring no proteins went to waste. The principle of maximized utilizations aligned well with wartime scarcity and post-war rebuilding efforts. The process yielded balanced outputs with no single proteins dominating. Fractionators pursued a broad portfolio aiming to meet diverse national and public health needs. This approach was also driven by self-sufficiency policies, practically in Europe and the US. The Kohn fractionations from 1 to 5 supported a wide range of therapeutic indications, from bleeding disorders to infectious diseases prophylaxis. Gamma globulins, for instance, were used to prevent measles and hepatitis in exposed populations. And albumin, of course, was king. It was the first protein ever fractionated at industrial scale, crucial during World War II for shock and volume depletion. Its stability and long shelf life made it the flagship product for early fractionation. This ecosystem, though noble in its intent, was deeply rooted in public health objectives and not necessarily in market value. This distinction becomes critical as we transition towards the next model. Here we introduce a co-tension in today's plasma strategy: health economies versus molecular prioritization. The radar graph at the center of this slide contrasts two distinct models: an IVIG dominant fractionation strategy and a balanced fractionation model that distributes value across the full plasma proteome. Let's break this down. From a health economic perspective, plasma scarcity, we look at precise resource allocation, budget impact forecasting, attention to the market volatility, and rigorous cost-effective analysis across all indications. However, IVIG, due to its high price point and demand trajectory, it's becoming the gravitational center of the industry, the whole industry. This has led to disproportionate channeling of plasma volume and capital towards IgG extraction, often at the expense of albumin, clotting factors, and niche proteins. This introduces challenges in plasma availabilities. Costs rise across the board, scarcity in non-IgG proteins worsens, and ethical dilemmas can rise and are rising in LMIC settings, where IVIG is not affordable. Yet albumin or factors like coagulation factors from 8 to 9 to etc. might be life-saving. Clinical outcomes become tethered to global production monopolies. Our goal as transfusion scientists is to ensure that value doesn't blind us to therapeutic diversity, especially when shortages in one molecule can result in avoidable morbidity or mortality elsewhere. Let me fix this. This slide visualizes a transformative economic shift in the plasma industry. On the left, we see the past model where it's the same model, actually. It's we're talking about the first liter and last liter model here. And we can see the past model, how this new model is applied during the past times, where albumin was cornerstone of plasma fractionation. It represented the majority of the yield, and until recently, a major share of revenue. This reflects the historic first liter logic, where plasma was processed from volume replacement and acute care use, particularly during wartime. On the right, the current model shows the new reality. Where albumin still represents a large yield, the inner circle, though, reveals a dramatic inversion. IVIG, once a minority player, now commands 70% of the total revenue. Albumin, despite being the most abundant fractionation, has dropped to only 10% of the revenue shares. Clotting factors and other remains constant in physical yield, but declining in commercial emphasis. This illustrates the last liter strategy, where the economic focus has shifted to extracting the most value per liter, often to determine the equitable protein distribution. For LMICs, what does this mean? This creates a critical challenge. Albumin and clotting factors are more critically relevant than IVIG in many public healthcare settings, and they're now commercially marginalized. This imbalance demands academic scrutiny and policy response. This slide captures the essence of the contemporary plasma economy, now driven by value, not volume. The key inflection point is the global surge in IVIG demand, which has reshaped operational priorities across the industry. The concept of last liter economies refers to how marginal revenue is extracted after core proteins yield are secured. Only immunoglobulins, in this case, remains economically attractive at scale. As a result, fractionators maximize IgG extraction, even if it means sidelining other value proteins. This shift has critical consequences. Other fractionators, such as albumin, antithrombin, or other clotting factors, are deprioritized, not due to clinical irrelevance, but due to commercial underperformance. Albumin, once the flagship of fractionation, is now treated as a byproduct, but stagnant and declining prices in many markets. It's produced in large volumes and often stockpiled, underutilized, or sold at cost. This reveals a deeper issue. Clinical utility and market value have diverged. Essential products are no longer prioritized if they lack economic pull, raising important ethical and economic and regulatory questions. This slide, let me do this. In this slide, we present two interlinked truths about the global plasma economy. On the left, you can see that this is the market trend line showing the entire PDMP market projected to grow from 35 billion nowadays to 80 billion by 2034. This isn't just a linear growth, it reflects a rapid acceleration driven by rising demand for plasma-derived medicinal products worldwide. Now to the right, you can see that there is a pie chart reveals the distribution of the market. The message is clear. Immunoglobulin holds the biggest pie chunk, and it holds it at approximately more than 60% of the total market revenue, towering above albumin, clotting factors, and other derivatives. This dominance makes IVIG the economic engine of the industry, the cash cow that defines how plasma is collected, processed, and priced. It also shapes corporate behavior, production strategies, prioritize maximizing IG yield, while other essential fractionations are increasingly treated as secondary output. In short, IVIG isn't just most profitable product, it's straining the entire direction of the plasma-derived medicinal product market. Let's go deeper in this. So the bar graph illustrates the retail price per gram per key plasma-derived medicinal product. At one end of the spectrum, at the left, you can see that the albumin, despite making the majority of the yield, is valued at approximately $5 per gram, and IVIG right next to it, you can see that it's approximately around $60 per gram. Already a dramatic jump. But the real economy outliers are the specialized protein over there. And we are talking now about factors, fibrinogen, antithrombin, and alpha-1 antitrypsin, which can reach up to $2,500 USD per gram. These prices underscore why companies chase certain fractionations, not because they yield the most in volume, but because they command exceptional value per gram. Yet this pricing asymmetry also creates tension. Therapies with smaller patient base but high price tags are prioritized. Essential but low-cost molecules like albumin are devalued and sometimes wasted. In sum, plasma isn't just biologically rich, it's economically tired, and that stratification is now driving fractionation strategy. This slide captures the tension of the core of the modern plasma economies, where market value overrides clinical breeds. Let's start on the left. This bar chart shows global annual cases treated by each plasma-derived medicinal product. Albumin, though modestly priced, treats around 16 million patients per year. Now, let me emphasize this. These numbers are coming from mainly from two continents, from Europe and US. This is not the whole global representation. The highest by far, IVIG, treats around 4 million around the world, and the rest, like coagulation factors, antithrombin, etc., reach only a fractionation of that. Most treat fewer than 1 million cases globally, and yet IVIG controls the market. Now this shifts. Sorry. Now this shift should be integrated in a chart that we can understand why is this happening. Shift your eyes to this chart over here. This pie chart tells us only 30% of the total demand is being met. Despite the urgent clinical need for volume-based products like albumin, IVIG drives the collection target and shapes the fractionation process. This economy imbalances stems from several factors. IVIG enjoys consistently rising demand in immunology and neurology and beyond. It commands high margin and it's immune to aggressive pricing and competition. Most importantly, IVIG is irreplaceable. Unlike albumin, where recombinant versions are already in trial phases, such in China, they are doing it from rice, immunoglobulins have repeatedly resisted the synthetic replication due to their molecular complexity, functional heterogeneity, and patient-specific targeting behavior. There is no viable recombinant IgG platform on the horizon, making plasma the only source. That biological exclusivity makes IVIG a non-negotiable output for manufacturers, even though albumin treats 4 times the patients. It's considered a byproduct, often sold at near cost or bundled with IVIG contract. So what we're witnessing in the consequences of the last liter economy, where the economic gravity shifts towards the product that justifies extracting every last drop regardless of public health parity. This slide reinforces the market asymmetry between two of the most abundant plasma fractionation, IVIG and albumin, and how this imbalance dictates behavioral change. On the left, we see IVIG, a high-value, irreplaceable therapeutic, used across immunology and neurology, with prices ranging from 50 to 100 grams per USD per gram. Even a single vial carries major commercial weight. The global IVIG market was established nowadays at 15 billion in 2023 and 2024, and projected to rise and double to 28 billion by 2030. In the middle, we see the albumin, a life-saving product, especially in volume-expanding therapies, burn treatments, and liver diseases. Yet, it trades at only 5 to 15 USD per gram. Despite its higher volume of use, it's economically marginalized, often labeled as co-product or byproduct, not a driver. On the right, the implication becomes clear. Production focus follows the money. Fractionators are incentivized to optimize processes for maximum IVIG yield per liter, often at the expense of other valuable proteins. This is what we call the value over volume paradigm. It's not about how many lives a product touches, it's about how much return per gram it generates. This price-weight strategy leads to lower investment in balanced fractionation, reduce availability of low-cost, high-use proteins like albumin. A supply chain built around IgG-centric economies, not public healthcare outcomes. And because no recombinant IVIG exists or is close to being commercialized, plasma dependency for IgG remains absolute. This gives IVIG an economic veto over the entire plasma processing pipeline. Now, this map will show us what's happening globally. Roughly 70% of all plasma uses comes from one country, the US. Even though it represents just 5% of the global population, this is primarily due to the paid donor model, which is largely restricted or banned elsewhere. Now, this isn't just an American story, it's a global liability. We saw this during COVID-19, where disruption in the US donor centers led to a global shortage in IVIG and other molecules as well, exposing the fragility of the entire supply chain. Furthermore, the commercial leadership of major players such as CSL, Takeda, Grifols, you name it, has reinforced a model where production capacity is centralized, but global needs are not equally served. The International Plasma and Fractionation Association Association, IPFA, and other stakeholders in the market are now calling for urgent diversification. That means expanding collection to regions like Southeast Asia, the Middle East, and Africa, improving structure and utilizing currently discarded recovered plasma to reduce waste. For companies like ours, this is more than a challenge. It's a call to action to rebalance the global plasma ecosystem and ensure therapeutic equity for all nations, not just those with collection centers. In this pie chart, we'll have a look at the clinical disparity in the global supply chain, and we are looking at everything. We're talking about source plasma and recovered plasma across the three dimensions: volume, donation frequency, and IgG yield. On the left, we see that the U.S. alone produces 72 million liters of source plasma annually, dwarfing the 18 million liters of recovered plasma that's being used globally. What's even worse and alarming is the 9 million liters of recovered plasma discarded every year in low and middle income countries. Now, this is based on a WHO document that was released and talking about the numbers that we have access to. We don't have access to everything, much like I'm just reminding you that we are talking about the major collection of data coming from two continents, Europe and the US, due to the lack of processing of infrastructure. That's an enormous loss of both treatment potential and the human effort. Let's look at donor frequency over here. Source plasma donors can give up to 104 times per year thanks to the regulated plasmapheresis model. In contrast, whole donations for recovered plasma is limited to about 6.5 times annually. But here's the twist. Recovered plasma actually contains more IgG per liter than the source plasma. So not only we are wasting valuable plasma, we are wasting plasma that's richer in therapeutic proteins. If we connect this to the earlier slides where we saw that IVIG driving the entire economy, this underlines the strategic blind spot in current models. Maximizing only source plasma ignores the untapped potential of recovered plasma practically and particularly in LM. The message here is simple. Recovered plasma is not inferior, it's underutilized. The future of sustainable and ethical plasma supply must integrate both systems, unlocking the full value of every donation. The case of India. In this case, we are first looking at all things. First, despite collecting over 10 million of units of blood annually, India sends only around 0.5 million liters of plasma for fractionation. That's less than 5% utilization. A massive disconnect between donation and therapeutic conversion. The local manufacturing is limited to just two products, IVIG and albumin, of course. Over 10 other plasma-derived medicinal products are entirely absent from the domestic portfolio, meaning patients are either underserved or dependent on imports. Self-sufficiency levels reflect this gap. Only 46% of IVIG demand is met locally. For albumin, the rate is even lower. Everything else fully imported, making the market highly vulnerable to global price shifts and supply shocks. But there is a real issue. India prohibits compensated plasmapheresis, which means source plasma collection is not feasible at scale under current regulations. Instead, the country relies on recovered plasma, a far less frequent, less optimized streamline. This landscape perfectly illustrates why structural reforms are needed, not just in investment. Policy updates enabling ethical source plasma collection could revolutionize access and reduce import reliance and position India as a global regional hub for PDMPs. Let's talk dilemmas. The US model is based on paid high-frequency plasmapheresis, whereas the EU or Europe mandates voluntary donations. While both models are medically safe, paid plasma carries a stigma, often seen as exploitative, especially when low-income donors are involved. This raises difficult questions. Are we saving lives or commodifying vulnerability? This shift to the market distortions. IVIG commands top priority in fractionation because of its profitability, often at expenses of albumin, hyperimmune, and rare diseases therapies. Hyperimmune globulins, though essential in epidemics and rare infections, receive little investment due to niche demands. Regional inequities compounds that issue. Wealthy countries can access comprehensive therapies portfolio, while lower income countries or nations face systematic shortages despite donating significant volumes of blood. Compare the regulatory differences. The FDA allows donations 104 times a year, incentivize a high throughput And the SOHO regulations in Europe, on the other hand, limit that to 64 times per year and only from unpaid donors. Let's stay here. So meanwhile, recovered plasma from transfusion services come at lower volumes but higher IgG concentrations, yet vastly underutilized. Beyond ethics and economies, there are critical regulatory and logistics hurdles that shape plasma access globally. Now we are talking about cross-border complexities. Plasma may be collected in one country, fractionated in another, and then sold globally. This fragmented model introduces regulatory gaps, increasing transit risks, and makes national supply chains vulnerable to geopolitical and trade disruptions, much like what's happening in the Red Sea, for example. Data transparency and gaps in many nations. We lack the accurate tracking system of plasma collection, utilization, and product wastage through clinical demand. Without this data, governments and health systems can't design sustainable or responsive plasma policies, and waste often goes unnoticed. And we are talking now about the contract of fractionations. The WHO recommends toll fractionation to build local self-sufficiency and it remains underutilized. But why? Regulatory constraints. We have a big regulatory constraint when it comes to every single nation. Lack of quality of plasma, absence of GMP compliant infrastructure, and limited international cooperation. All of these factors hinder equitable plasma access, especially in LMICs. This message here is clear. Fixing plasma procurement is not just about money or technology. It's about transparency, regulatory alignments, and collaborative governance. Only then we can truly close the global plasma gap. The direction of the global fractionation is shaped by five covering forces. It's critical we balance innovation with equity. The global supply remains fragile because collection is over-concentrated in the US. To fix this, we must expand voluntary plasmapheresis in LMICs and build systems that fractionate recovered plasma, which is currently underutilized. This expands access without relying on compensated donation models. WHO's national plasma strategy is the cornerstone, but it's not enough. We need sustained public-private partnerships to fund fractionation capacity, especially in countries like India. The goal is long-term independence, not perpetual import reliance. Beyond immunoglobulins, albumin is emerging as a value-added molecule with applications in drug delivery, nanoparticles, and tissue engineering. Plasma fractionation must be evolved to unlock these lower yield, high impact proteins. And talking about technology, the dominance of IVIG has pushed towards breakthroughs in purifications, especially chromatography-based techniques that increase Ig yield and safety. A defining example is the isoagglutinins removal. High titer of anti-A and anti-B antibodies are proven to have caused many mismatches when we are delivering these drugs. In IVIG, caused hemolysis in most cases. Manufacturers now use immunoaffinity chromatography to filter these out. And the Ig isolo columns, for instance, reduce anti-A titers fourfolds. And this showed results that are very promising. More than 95% drop in hemolytic reactions in reports. This has made high-dose IVIG infusions safer, especially in regions with diverse blood groups, a clear case of innovation pulled by clinical necessity. So, the blood plasma was once discarded and wasted. Today, it's recognized for what it truly is, a strategic resource. A global scarcity is now pulling everything together to secure access. The world didn't shift to value because of innovation. We were dragged into it by scarcity. Supply shortfalls, donor dropouts, and import dependencies have forced a rethink of how plasma is collected, processed, and prioritized. And yet, despite the urgency, investment appetite in plasma remains weak. This niche critical industry still lacks the attention it deserves. So yes, we need balance. Align financial models with public health outcomes, making sure we fund all plasma-derived medicinal products, not just those promising, much like IgG revenues. For us in Transfusion, this is a call to slash plasma wastage, close data gaps, advocate national self-sufficiency, and elevate plasma to the next level of strategic sovereignty. This isn't just a biotech challenge. It's more of a moral collaboration for a whole system. The only metric that should guide us is patient lives, not quarterly returns, not IgG volume curves, just lives. Thank you.
Thank you,
Thank
sir.
you very
Linda
much.
Madam.
Yeah. Thank you, sir, for that thought-provoking and deeply informative talk. Now the session is open for discussion. Sir, if what are the serological markers which usually you check while after procuring the plasma components?
Usually everything is done practically much like we do in blood banks. The first markers that we are looking for are, of course, the serological criteria, and of course, every place we are operating, we take into consideration any local endemics that's happening. So we might add several serological tests. For example, once in Lebanon, we added the VDRL, not the VDR, the HTLV test, because we thought that a pandemic was happening or local endemic was happening in Lebanon. And we, of course, look for the protein levels on all matters. We're looking at the total proteins, and of course, we look at the albumin level. Now, there was a big question mark whether we should go for NAT technology or we should go for let's say inactivation. But since we are partnering with the global fractionators, they are handling this on their side. So they are inactivating all 4 viruses, and they are doing the NAT technology on the pools themselves.
Okay, sir. One more question. Each country will not be having this testing the same infectious serology markers. So each country will be having separate markers will be there. So how you do that? How you perform it your side?
You need to repeat that. I barely heard you. Sorry.
Sir, for India, for example, we'll be screening only for hepatitis B, HCV, and HIV, and we test for malaria and syphilis. But for other country, it will not be the same. So how you do that?
We
At your
adjust.
center?
We adjust our yes. We adjust our testing model. It's all about an analyzer, and it's all about what we want to intend to do or what we want to test for. If I'm operating, for example, in I don't know, if we are operating in Africa, for example, it is so important for us to test for malaria, for example. If we are testing by the end of the day, remember one thing: uh, opening a plasma collection center or an activity in a country where the donation pool is untapped. This opens a lot of margin, and we are looking as well for like things for natural formation of natural antibodies or recurrence. Like for example, in one area that we were investigating, there was a high level of rabies. And we were looking into whether some of the donors were hyperimmune for rabies, in order for us to select and collectively try to segregate the plasma in a way where we can send this plasma for fractionation for hyperimmune anti-rabies PDMP. So it's totally dependent on the area where we are operating in. And if in case we need to add tests or remove tests or change whatever we are doing, it's totally facultative and totally can be done.
Okay, sir. And one more question. What are the ethical and regulatory considerations when sourcing plasma internationally, especially from paid donors?
Well, much like you've seen in the presentation, there are 2 regulatory bodies out there that are governing everything that's happening with the paid and unpaid version. We're talking about the FDA that's happening in the US. Although
Okay.
although it's it although we are talking about a loose system, let's call it, where everything is possible and you can do whatever. It's purely commercialized, and we are talking about the SOHO regulations that are implemented in Europe. Although we have like several countries in Europe that already broke free from that system, because they much like you've seen in the FDA governance of things, you can see that we are not only the US is supplying plasma for itself, is supplying plasma for the world, and this system proved that it is very effective. But on the other hand, there are some ethical issues when it comes to how many times this donor can come, how many times, or what should we be testing when it comes to this donor. And when we go to Europe, we see this drastic drop to 64 donations per year, and they change to an altruism approach. They want all donors to come and donate out of the blue. Now, if we're talking about that, it's proven that this system is very fragile, because by the end of the day, most Europe is reliant on paid donors that are coming from the US.
Okay, sir. Thank
This
you.
is the actual dilemma.
No, Pooja, is there any other questions from the participants? Any
Yes, ma'am.
questions
There
from-
is a There is a question in the chat box. It's asked by Dr. Harpreet Singh. He has asked, what is the experience with other developing countries, especially the Southeast Asia and African countries?
Let me check this. Sorry, can you repeat the question? I'm trying to check it in the chat as we speak.
Sure, sir. Dr. Harpreet Singh has asked, what is the experience with other developing countries, especially the Southeast Asian and African countries?
Well, yes. Let's talk about this. Building capacity in these two regions is our top priority, because if we want to look at what's available from the data out there, we can see that all right. In 2019, when COVID-19 hit, there were warnings that were issued in Europe, for example in the UK, and we are talking here about an era before they started using their own plasma because of Creutzfeldt-Jakob disease. In 2019, when the shortfall hit, it hit all markets. But the problem is that in low, middle, and income countries, we had death tolls. In Europe, because they had strategic reserves of mainly all the molecules, all they did is issued warnings and changed the practices. Now, this tells us a lot. This tells us first that whatever let's say data that's coming, it's coming from majorly two points in this world, while more than 60% of the world is left in a blind spot, much like Southeast Asia and the African countries. Now, when we are talking about Africa, we're talking about a waste that's not even counted. We're talking about capacity that's not even there. So what's the main target for us? It's to build sustainable and ethical and standardized capacity throughout all the African continent, so as much as we are intending to do this in Southeast Asia. And then we would like to, as much as possible, collaborate on getting as structured data and trying to, as much as possible, link these nations or where we are operating into the global network. Now we are talking about the plasma master file, and we are talking about whatever these big fractionators are doing in a collaborative movement. Now we want to integrate every single nation into that plasma master file, and how the global landscape is being shaped when it comes to the plasma-derived medicine. To answer your question, we don't really know. We are trying to find out.
Thank you, sir. There's another question by the same doctor. Just for understanding, paid or donor compensation for time and logistics. If it is donor compensation, then India recently modified its policy and included in donor compensation. The second point is plasma price is too low to be easily misused even for albumin replacement.
Well, yes. Much like where we are trying to position a system that can actually look at both sides. My expertise come from low and middle income countries. I operated mainly in the Middle East, and I know that it is very tough for us to understand whether we should go for and follow the FDA guidelines or follow the SOHO guidelines. There is a big difference. So if I want to look at things now, the best way to do this is to establish first a framework where both sides can be tackled. Now we don't need to go like, for example, in the US they go for up to $80 per donation. Now this would attract donors and would outsource donors maybe from donating blood or donating plasma, especially in rural areas. Now where we go into the low and middle income countries, we are talking about a population that's in majority can be suffering from this low income. Now, how do we approach this? We need to raise awareness. We need to publicly state that we are doing, let's say, this kind of service. There are so many ways to remunerate and retain a donor, not directly through finances, if you know what I mean, what I'm talking about here. Now, if I want to position a system where we can actually drive these compensations, we need to look at both sides. We need to try to tailor whatever is happening at the global scale in a bipolar system to whatever we think is logical and ethical in every single country we are working with. That's why we collaborate with the Ministry of Health. We totally understand their healthcare needs. And by the end of the day, if we can find a system and this system is within two frames, not just one, that can save the lives in a country we are operating in, this is where we are going.
Thank you, sir, for answering that. We have another question by Mr. Ashish. He has asked, do you find any difference in quality of recovered plasma in different countries?
Well, the experience from one country to one country differs a lot. Now, for example, in Lebanon, where we established kind of a standardized system, you can see that, or you can actually feel that every single bag of plasma that's coming from the blood banks is approximately the same volume. Now this tells you a lot. First of all, we have the same labeling technology. We have the same, let's say, ISO guided and let's say certified guided way of doing things, which is the GMP practices when it comes to the blood bank and when it comes to the fractionation. Now, this tells you a lot. We are in a framework where every single part of the country is supplying the same quality and same volume, approximation of volume in a bag of plasma. Going to other countries, I'm gonna share this experience. I was talking with somebody from India, and the doctor was telling me that I received once a bag approximately 250 mLs of plasma in it, and another bag that was as big as a football, let's say. And I was like, why is that happening? Now, because of the drift in the volume. Now we are talking about a volume that's completely translating into value. And people would think that with more volume, it doesn't matter how they are doing it or when they are doing it. This bigger volume will get them more income. Now we need to talk about this and how to standardize this and how to make it patient safe and fractionation safe. And the only way to do this is to standardize the practice, of course, with the collaboration of the Ministry of Health. No other way.
Thank you, sir. Next question is from Mr. Nitin. He has asked, with the global crisis on plasma product manufacturing, what could be the main challenge if India has to import the plasma, as plasmapheresis is not allowed in India?
Well, plasmapheresis is not allowed. Plasmapheresis is allowed everywhere in the world. It's a technology. It's proven its efficiency and efficacy. Now we're talking about a device that allows the donor to donate more than let's say more than once per 2 months, much like in blood banks. Now the big dilemma is not allowing the plasmapheresis, is how to handle the plasmapheresis in ways where we can actually let's say, compensate this donor for coming and donating for the industry without, let's say, going over the borderline or the cross line where we say it should be unpaid and altruistic. Now, if I want to look at this way, much like I said, we need to tailor our own operative system, and we need to standardize the system across all the country, and we need to collaborate specifically with the Ministry of Health in order to get this done properly and ethically, and procure plasma from both sides, from plasmapheresis side as sourcing, and from the blood banking side as recovered plasma, but both done ethically and in a proper way.
Thank you, sir. We have one more question. It's from Aitno Atnaf. I hope I'm pronouncing it right. The question is, in Africa, particularly in Ethiopia, it is impossible to achieve this. But as an immunohematologist, I have obtained new techniques that I should share with my students.
Yes, let's talk about this. The main problem with driving this forward is the willpower, and on top of the willpower is the investment. Much like I said much like I mentioned in the presentation, the big problem is money. The big problem is when and how and where to procure this big amount of money in order to establish this standardization everywhere and establish this technology everywhere. Building infrastructure is not simple. It's multi-layered, complex, and we have to work with every single person that's integrated somehow in this industry. We're talking about everyone. We're talking about people that work in construction, people that work in the blood banks, people that work in the pharmaceutical sector. We are talking about everyone. Now, bringing the money in will let us have the power and have the will to build this on a national scale. We've done it already. When we first had a look at how things were in Lebanon, for example, the situation was even worse than Ethiopia, trust me. But by the end of the day, because we could bring the money in, and because we had the willpower and the knowledge on how to do things, we achieved the impossible. It's that simple. We've done it. Once we have the money, once we have the willpower and the people to do it, there is nothing stopping us. That's to answer the Ethiopian situation.
Thank you, sir. Now we have one more question from Dr. Harpreet Singh. He's asked, in the US, blood services are fragmented, whereas in Europe, they are centralized or regional. How should health systems approach for the Indian scenario, which is fragmented, but the approach is like a centralized one?
Exactly. Again, I'm gonna refer back to Lebanon. When we started working on this back in the days, we were talking about a centralized system that's being born. It was governed by the Lebanese Red Cross back then, and they had the vision of creating these centralized blood banking, let's say, services all around the countries. And what we had before that is familial blood bank. So we are talking about a hybrid system that already been existed in one way or another, and we wanted to achieve this on all sides, on all levels. Now, if you look at how things are done in Lebanon, we have approximately 13 blood transfusion centers that are already centralized by the Lebanese Red Cross, and you could see on the other side practically all university hospitals that house a familial blood bank in them have the same quality as what's happening in the centralized blood bank. Now, we are not saying that we should pivot one system into the other system. What we are saying is that we can build capacity on both, and we can standardize a framework that can accommodate both. And this is our approach. We want to work whatever that is, tailor whatever that is, and make functional whatever that is. We are not changing practices.
Thank you, sir. I don't think we have any more questions. Linda, ma'am, do you have anything else to add?
Sir, I have one more question. Usually, what is being done during the production phase? You have already mentioned to reduce these blood group antibody titers, especially in case of IVIG, because as you said, this can, if the titer level is high in case of IVIG, it can cause many serological problems. So while in the manufacturing phase, do you do anything to reduce these titers?
No, actually, we are not diving into this because I was once asked why aren't we going into fractionation and going into purification as we speak? Well, this is it. We do have the money, we do have the funding to establish fractionation and to establish purification technologies, but looking at things, we would be like burning billions on an engine that doesn't have a fuel to run. What we intend to do is first change the way things are. We want to procure ethical plasma from all sides. That's one. Two, when we are talking about the technologies out there are some companies already that are investing in like resin technology in order to absorb all these let's say impurities when it comes to the IVIG injections. There was a nice leap towards an incremental market share when it comes to this technology, because much like I detailed, it's the whole industry is being driven by the IVIG molecule now. As long as we are increasing up, this it will still let's say train whatever is happening in this technology and similar technologies to be more prominent in the market. This is being done. And when it comes to the fractionation itself, we don't intend to open any fractionation as we speak. We're not even diving into the concept of fractionating a plasma as we speak. What we wanna establish first is let's say remove this blindfold or blind spots from around the world. We want to integrate all nations into this plasma industry before we start thinking, okay, now India, for example, this is my personal opinion in this. One of the biggest countries and the most populous countries in the world should have its own fractionation, and we're talking about a large-scale fractionation. We're talking about all molecules, not just IVIG, not just albumin or whatever. In order to achieve this, we need to standardize the plasma procurement. This is it.
I think according to FDA, they mentioned that the anti-antibody titers in the IVIG should be minimum cutoff. They have mentioned as 64. So usually, what we perform in the fractionation unit is that the chromatographic method. But I think they have limitations to restricting the anti-AB antibody. They limit only the level of anti and anti-B titers. Isn't that so?
Yes, it is true. It is true. They have a limitation when it comes to the injection itself and the dosage and the dosage effect. Yes, they are looking into this, and there are some studies that showed even though we are practically all manufacturers are reaching this below that level, that's 64. But the problem is that even though they are doing this, some injections are developing hemolysis. And this has been a big problem. That's why there are you would see there is no regulation when it comes to this, but you would see that there is this global shift towards focusing on this technology, and many fractionators already adapted this technology in order for them to get rid of this anti-A and anti-B noise that's present in the IVIG solutions. So that's the main. That's why I let's say talked about this technology.
Okay. Thank you, sir.
You're welcome.
So, any more questions, Pooja? If no more questions.
No more questions.
Okay. And once again, I would like to thank you, sir, for sharing your time, experience, and vision with us today. And thank you to all the participants for your kind attention and engagement. So, without any delay, we would conclude today's session. Thank you all. Thank you once again.
And thank you all for being here.
Yeah, so from TIMAS, a sincere thank you to our speaker, Mr. Antoine Francis, for their extremely informative presentation. And thank you, Linda ma'am, for chairing the session. We also thank our participants for being with us today. We have shared a feedback link in the chat box. Please do fill it to receive a participation certificate. And we will soon make a recorded session of the webinar available on our YouTube channel. Stay connected with TIMAS for more such academic engagements. Have a great day ahead. Thank you.
Thank you all.